What does it mean to be alive? That's a simple question, but the answer is not simple at all. A rock is not alive. We know that. A dog is alive. We know that too. But what about a virus? It can't do anything on its own. It needs to hijack a living cell to make copies of itself. So, is it truly alive, or is it just a complicated piece of non-living machinery? The line between life and non-life gets blurry the closer you look, but for the most part, we agree on a few basic rules. Living things have a boundary, something that separates inside from outside. For cells, this is a membrane. Living things have a metabolism. They take in energy and materials from the environment and use them to build themselves and stay running. And living things carry information, a blueprint. In modern life, this is DNA. This blueprint allows them to reproduce, passing that information on to the next generation. And crucially, this system can evolve. It can change over time. So here's the big question. Can we take things that are clearly not alive, like simple chemicals in a bottle, and assemble them into something that is alive? Can we build a living cell from non-living parts? This isn't about cloning. Cloning starts with a living cell. This isn't about genetic engineering. That's just modifying a living cell that already exists. This is about starting with the fundamental building blocks, the molecules, the carbon, hydrogen, oxygen, nitrogen, and phosphorus. The stuff you can buy from a chemical supply company, and from that chemical soup, coaxing a new simple form of life into existence. It sounds like science fiction. And for a long time it was, but now it's a real field of science. It's called synthetic biology, or sometimes origin of life research. Scientists around the world are trying to do exactly this. Not to create a complex modern bacterium like E. coli, that would be like trying to build a supercomputer on your first day learning about circuits. The goal is much more modest. The goal is to build a protocell, a simple, primitive cell that just barely meets the criteria for being alive. A bubble that can hold some information, run a simple chemical reaction for energy, and maybe, just maybe, make a sloppy copy of itself. If we could do that, it would change everything. It would show us that life isn't a magical spark. It's a process. It's something that can emerge from chemistry and physics if the conditions are right. It would be one of the most profound discoveries in human history. So, how close are we? To understand the challenge, we need to break it down. We need to build life's three essential components from scratch and get them to work together. The container, the blueprint, and the engine. First, you need a container. You can't have a cell without a wall. Without a boundary, all the important molecules inside would just float away. The cell's contents would mix with the ocean or the pond, and that would be the end of it. Life needs to concentrate things. It needs a special, protected environment on the inside where the chemistry of life can happen. In all known life, this container is the cell membrane. It's made of molecules called lipids, specifically phospholipids. These are fascinating molecules. They have a head that loves water and a tail that hates it. When you drop these molecules into water, they do something amazing all by themselves. They self-assemble. The water-hating tails all try to hide from the water, clustering together. The water-loving heads arrange themselves to face the water. The most stable way for them to do this is to form a little sphere, or a bubble. This bubble is called a vesicle, and the wall of this bubble isn't just a single layer. It's a double layer, a bilayer. The tails face inward toward each other, creating an oily, water-free zone. The heads face outward, one layer toward the water outside the bubble, and one layer toward the water trapped inside the bubble. This is the basic structure of every cell membrane on Earth. The fact that these vesicles form on their own is a huge gift from chemistry. It's one part of the puzzle that nature seems to solve for us. Scientists can now make these vesicles in the lab with ease. You take some lipids, add water, shake it up, and you get billions of tiny empty protocells. You have your container, but a simple container isn't enough. A real cell membrane has to do more than just sit there. It has to be able to grow. If the cell is going to divide, its membrane needs to get bigger first. Researchers are working on this. They found that if you feed a vesicle with more lipids, the new lipids can insert themselves into the existing membrane, making the vesicle grow. Under certain conditions, these growing vesicles can even get long and stringy, like a noodle, and then break into smaller daughter vesicles. It's not the neat, orderly division of a modern cell, but it's a form of physical reproduction. So, step one, the container, is looking pretty good. We can make them, and we can get them to grow and divide in a primitive way. But an empty container is not a living cell. It's just a bubble. The next part is the real challenge. You need a blueprint. You need information, a set of instructions that tells the cell what to do and how to build its parts. In modern life, that blueprint is DNA. DNA is an incredible molecule. It's a long, twisted ladder, a double helix. The rungs of the ladder are made of 4 different chemical letters, A, T, C, and G. The sequence of these letters is the genetic code. It's the recipe book for life. A section of DNA, a gene, holds the instructions for building a specific protein. Proteins are the workers of the cell. They are the enzymes that speed up chemical reactions, the structural beams that give the cell its shape, the motors that let it move. Here we run into a classic chicken and egg problem. DNA holds the instructions for making proteins. But you need proteins, very specific enzymes, to read the DNA and to make copies of it. So which came first? The DNA blueprint or the protein machinery needed to read it? It's a paradox that puzzled scientists for decades. The solution might be another molecule, a close cousin of DNA called RNA. RNA is similar to DNA, but it's usually single-stranded, not a double helix. And it uses a slightly different set of chemical letters. In modern cells, RNA has many jobs. It often acts as a messenger. A copy of a gene is made in RNA, and that messenger RNA is then carried to the cell's factory, the ribosome, where it's used as a template to build a protein. But some scientists think that in the very first life on Earth, RNA did everything. This is the RNA world hypothesis. The idea is that RNA was both the blueprint and the machinery. It could store genetic information just like DNA, but because it can fold up into complex 3-dimensional shapes like a protein, it can also act as an enzyme. These RNA enzymes are called ribozymes. This discovery was a game changer. It solved the chicken and egg problem. In a hypothetical RNA world, you don't need DNA or proteins to get started. You just need RNA. An RNA molecule could carry the instructions for making copies of itself, and it could also be the machine that does the copying. So, the goal for synthetic cell builders became clear. They needed to get a self-replicating piece of RNA inside one of those lipid vesicles. This has proven to be incredibly difficult. First, you need an RNA molecule that can copy itself. Scientists have had some success with this in a test tube. They've used a form of artificial evolution to design ribozymes that can stitch together a new strand of RNA, using an existing strand as a template. But the process is slow, clumsy, and makes a lot of mistakes. It's a long way from the efficient replication we see in modern cells. Then you have to get this whole system to work inside a tiny vesicle, and you need to get the building blocks for RNA, the individual chemical letters, to cross the membrane and get inside where they can be used. This is a huge problem. That lipid membrane that's so good at keeping the important stuff in is also very good at keeping the building blocks out. Some labs have found that adding certain minerals to the mix, like magnesium, can help the RNA chemistry. Others have found that if the vesicles are constantly going through wet and dry cycles, it can help concentrate the molecules and get them across the membrane. Progress is being made, but it's slow. Getting a piece of genetic material to reliably replicate inside a membrane-bound compartment is perhaps the single biggest hurdle in building a synthetic cell. There is another way to think about the genetic blueprint. This is the top-down approach. Instead of building from scratch, you start with a living organism and strip it down to its bare essentials. This is the work pioneered by scientists like Craig Venter. His team took a very simple bacterium, one with a small number of genes. They then set about figuring out the absolute minimum set of genes required for that bacterium to live and reproduce in a lab dish full of nutrients. They did this by systematically knocking out genes, one by one, to see which ones were essential. After years of work, they had a blueprint for a minimal genome. It was the smallest set of instructions for life that they could design. Then they did something remarkable. They synthesized this minimal genome from scratch, from bottles of chemicals. They built a complete 1,000,000-letter-long DNA chromosome in the lab. Then they took another living bacterial cell, removed its own DNA, and transplanted this new synthetic DNA into it. The cell booted up. It came to life running on this new artificial software. The cell and its descendants were a new synthetic species defined by a genome, designed by a computer, and built by humans. This was a landmark achievement, but it's important to understand what it was and what it wasn't. It wasn't building a cell from non-living parts. They took a pre-existing living cell and gave it a new brain. The container, the machinery, the energy systems, all of that was borrowed from a living ancestor. It's more like swapping the operating system on a computer than building a computer from sand. Still, this top-down approach is incredibly valuable. It teaches us what the essential parts list for life is. It provides a target for the bottom-up builders to aim for. So we have the container, the vesicle. And we have a plan for the blueprint, either a simple self-replicating RNA or a minimal DNA genome. But there's a third crucial piece of the puzzle, the engine, the metabolism. A cell needs energy. A bag of replicating RNA is just going to sit there unless it has a power source. Life is work. It's the constant work of building, repairing, and copying. And work requires energy. In your body, you get energy by eating food and breathing oxygen. Your cells break down sugar molecules in a controlled burn, capturing the released energy in a special molecule called ATP. And ATP is like the cell's rechargeable battery. It carries energy to wherever it's needed in the cell. Plants get their energy from sunlight, using photosynthesis to create their own sugars. A protocell would need some kind of metabolism too. It wouldn't need the complex, multi-step metabolic pathways of a modern cell. It would need something much simpler, maybe just a single chemical reaction that releases energy. And critically, it would need a way to couple that energy to the important tasks, like copying its RNA. This is another huge challenge. How do you get the energy from reaction A to power reaction B, all within a microscopic bubble? In modern cells, this coupling is managed by a whole suite of specialized protein enzymes. A protocell wouldn't have those. One idea is that the first life didn't use complex chemical reactions, but instead harnessed simple physical gradients. Imagine a protocell living near an underwater volcanic vent. Outside the cell, the water might be acidic, full of protons. Inside, it might be less acidic. This difference, this proton gradient across the membrane, is a form of stored energy. It's like water behind a dam. Modern cells use proton gradients all the time to generate ATP. It's fundamental to how we get energy. Researchers are trying to see if they can recreate this in their lab-made vesicles. Can they create a simple gradient across the membrane and then use that energy to drive the formation of RNA or the creation of more lipids? Some early results are promising. They've shown that simple fatty acid vesicles are more stable in certain pH conditions and that they can harness a proton gradient to grow. It's a long way from a full metabolism, but it's a start. It suggests that the earliest forms of life might have been powered not by a complex internal engine, but by simply tapping into the energy of their environment. Now comes the hardest part of all, putting it all together. This is the integration problem. It's one thing to build a vesicle in one test tube. It's another thing to get RNA to replicate in a second test tube. And it's a third thing to create a simple energy-releasing reaction in a third test tube. But a living cell is all of these things happening at the same time in the same tiny space, all working together in a coordinated way. This is where the dream meets the brutal reality of chemistry. The conditions that are perfect for building a stable membrane might be terrible for replicating RNA. The metal ions that help the RNA enzyme work might destroy the lipid membrane. The energy-releasing reaction might produce byproducts that poison the whole system. Everything has to be compatible. The parts can't just coexist, they have to interact. In a truly living system, the blueprint would contain the information for building the container. The RNA might code for a ribozyme that helps synthesize more lipids, making the membrane grow. The metabolism would have to produce the energy needed to copy the RNA and also produce the chemical building blocks for both the RNA and the lipids. The system needs to pull itself up by its own bootstraps. It needs to become autonomous. It must maintain itself, build itself, and copy itself, all using materials and energy from its environment. This deep integration of components is what makes life so different from just a complicated bag of chemicals. A car is made of many parts, but if you grind up a car into dust and put it in a box, it will never reassemble itself. A living cell, however, is constantly reassembling itself, moment to moment. So, where are we now? Can we build a living cell from non-living parts? The honest answer is no, not yet. We have not created a new form of life in the lab from scratch. No one has built a system that combines a container, a blueprint, and a metabolism into a self-sustaining, replicating, evolving whole. But we have made incredible progress. We have the pieces. We can create the vesicles that form the container. We can get them to grow and divide. We have developed RNA molecules that can copy themselves, providing a plausible blueprint. We have built entire genomes from chemicals and used them to reboot existing cells. And we are beginning to understand how the simplest forms of metabolism might have worked by harnessing energy from the environment. The current frontier of research is focused on that integration problem. Labs are trying to get all these different systems to work together in the same vesicle. One group might be trying to get an RNA replicator to work inside a growing, dividing vesicle. Another might be trying to couple a simple light-capturing pigment in the membrane to the production of RNA building blocks inside. It's a step-by-step process. The first synthetic cell will not look like anything alive today. It will be fragile, it will be inefficient, its replication will be messy and full of errors. It will probably only be able to survive in very specific, carefully controlled lab conditions. It will barely be alive, but it will be a start. And if we succeed, the implications are staggering. On a practical level, it could open up entirely new fields of biotechnology. We could design and build cells to do specific jobs. Cells that produce clean fuel, cells that synthesize complex medicines, cells that detect and destroy cancer cells in the body. It would be the ultimate form of engineering. But the real impact is deeper. It's philosophical. For all of human history, we have seen life as something special, something unique, perhaps something divinely inspired. The origin of life has been one of the great mysteries. If we can show that life can be created from non-life just by following the laws of chemistry and physics, it will place humanity in a new context. It will tell us that life is not a miracle. It is a natural outcome of a complex, energy-rich chemical system. It will suggest that life might not be a rare accident confined to our planet. If life can arise from simple chemistry here, it could arise on any other planet with similar conditions. We might live in a universe that is teeming with life because the emergence of life is simply what happens when you leave the right chemicals cooking for long enough. The quest to build a cell is not just about a technical challenge, it's about understanding our own origins. It's an attempt to replay the tape of history, to figure out how the first living thing on our planet came to be some 4 billion years ago. We are trying to understand the bridge that connects the dead chemical world with the living biological world. The work is slow, it's frustrating. There are many dead ends and false starts, but every small success, every tiny step forward gets us closer. We have the basic Lego bricks of life. We are learning how they fit together. We haven't built the final structure yet. We don't have the complete instruction manual, but for the first time in history, building a living cell from scratch feels like it might just be possible. We are at the very beginning of finding out.